Hello, my name is Professor Nicholas Embleton, and I am a professor of neonatal medicine based in Newcastle upon Tyne in the north of England. And I'm going to be speaking today on probiotics in the prevention of necrotizing enterocolitis and other conditions in premature infants. And I'd like to start off by thanking the organisers for inviting me to speak to you today. So at the start, I think it's important just to disclose any conflicts that I have. Most of my research work has been funded by the National Institutes for Health Research or medical research councils or charities. I've also received institutional funding from industry, including Prolacta Biosciences and Danone Early Life Nutrition, but I don't receive any personal payment from those companies. I have accepted a lecture honoraria for this meeting today, but I will donate that honoraria to a neonatal research charity that I organise. I haven't got any shares, stock options. I have no financial conflicts with the probiotic industry, but I do provide non-remunerated advice to Neobiomics, who are a European company who market a probiotic product. So as I say, I'm from Newcastle upon Tyne in the north of England. You can see here the beautiful city we have with the River Tyne flowing through it. We're famous for a beer called Newcastle Brown Ale. We have a football team that never wins any trophies or the league ever, but we're still very passionate about them. And we have a beautiful countryside with lots of historic castles. From a research point of view, I've been involved in many large collaborative randomized controlled trials. Our research on our neonatal unit focuses very much on the microbiome and the metabolome. We're very interested in breast milk composition, particularly the role of IgA, but also the bacteriophage viruses. And we've also done work looking at human milk oligosaccharides. So a lot of our work is focused on necrotizing enterocolitis, immunology. And as part of that, we have a series of enteroids that are worked on by Dr. Chris Stewart here. So these are laboratory models grown from salvaged gut cells in preterm babies. So if we have a preterm baby who unfortunately requires an operation for NEC, we take parent consent to retrieve some of the resected tissue. And from any bits of live tissue that have been left, Chris is able to grow models in the laboratory, which represent the premature gastrointestinal tract. Some of our research has also followed the children up into early adulthood, where we've looked at long-term metabolic outcomes. And we also work closely with the Great North Neonatal Biobank, which is managed by my colleague, Dr. Janet Berrington. And in this, we have samples of stool, urine, salvaged blood, breast milk, and other tissue samples, which we collect with parent consent. And we now have tens of thousands of samples from over 1,000 preterm infants. So that's a bit of background in terms of my research program and interests. Now, of course, probiotics really should be seen as a component of nutrition. And therefore, I want to emphasize that nutrition is more than nutrients. Of course, as neonatologists, we focus on the importance of protein intake and micronutrients. But we're also now increasingly aware of the importance of what you might call functional components or bionutrients. And these include your human milk oligosaccharides, growth factors, IgA, enzymes. The next component of nutrition are the microbes. And these may be microbes within breast milk or promoted by breast milk, but also the critical role of the neonatal unit environment, and as we're focusing on today, the role of probiotics. And then the final component of nutrition are what I call the technical and socio-behavioral aspects. And these are very important, but these may include topics such as whether you should bolus feed or continuous feed, the role of skin-to-skin kangaroo care, sensory aspects of nutrition, behavior of healthcare staff towards mothers. So nutrition is very much more than nutrients. And over the last 20 years, as we have got better with our nutritional practice, we have seen survival in these infants improve. Of course, it's not just nutrition, but nutrition is a key component of our better outcomes. But as more babies now survive, the nutritional needs become even more important and more complex. All of us are looking after babies who may have developed NEC or sepsis, and feeding these babies is even more complicated. And it's important to note that many of the reasons why preterm babies have a complicated neonatal care are related to nutrition. So chronic lung disease, neurological complications, sepsis and NEC are all related to nutrition. So gut health links nutrition to many of these key morbidities. And as I say, as we get better at helping these babies survive, the importance of nutrition and microbial health increases. This is data, survival data from our unit. This is now a few years out of date, but I'm sure many of you will see a similar picture, which is that at all gestations less than 30 weeks, survival has increased. And remarkably, now in many units in high resource settings such as Europe and North America, we see survival of around 50% at 23 weeks gestation. And one of the questions is, what are the reasons for the other half who don't survive? And when we look at the causes of death, what we can see is that more than half of those babies who die from NEC or sepsis. And when you look at it in childhood terms, in high resource settings, more babies now die from NEC and sepsis than all cases of childhood cancer combined. And NEC and sepsis, as I mentioned, are closely linked to the microbiome. So let's have a little think about the microbiome and the bacterial world that we live in. Here I depict, well, from a publication, depict the whole of life on the earth as a month of 30 days. And you can see that in the first few millennia, there were no life forms on the planet, but then we developed prokaryotic and eukaryotic cells. And so throughout all of this time, the world is populated by viruses and bacteria, and humans only appear here on the very last day, the very last hour. So most of world evolution took place with only microbes present, and animals and plants only appear in the last few days of the month, and humans on the last day, the last hour. And then when you think about premature babies, of course, evolution never prepared a premature baby for early delivery. And we think of it from a genetic point of view, we can see that about 65% of human DNA evolved from microbes. Several studies over the last 20 years have shown how the pattern of bacteria in the gut change over time. So the pattern of bacteria in a newborn baby in the first week of life is very different to an adult. So you know that infants have a high proportion of bifidobacteria promoted by breastfeeding. And other bacteria such as Clostridia or Eubacteria, relatively low in early life, but increase as we get older. So this is the normal, natural pattern of gut colonization in humans. And where do babies acquire their bacteria from? Well, the bacteria are acquired from the mother's skin, the mother's genital tract, but also acquired from breast milk. And then the babies come home and they acquire microbes from their environment. And what you see is that in the first few days, the majority of the microbes are anaerobes, particularly bifidobacteria and Bacteroidetes. This is what you see in term infants. And many of these bacterial taxa are considered pioneer species. They get in there early. They create and sustain a low oxygen environment. And breastfeeding then interacts with this so that there are successive waves of colonization. But we know that in preterm babies, the biome, the microbiome, is very, very different. So in the first few seconds, the exposure to microbes for many preterm babies is profoundly different to what it is in term babies. Around half of our premature babies might be born by cesarean section. And then in the first few minutes, they come into contact with quite an abnormal environment, a sterile, clean environment, touched by people with clean rubber gloves. They're placed in incubators, they come into contact with other clean materials. And then on the neonatal unit, they live in a plastic box. So the environment of a preterm baby is completely different to that of a term baby. We then ensure that they're only touched by hands that have been washed in soap or gelled with alcohol or chlorhexidine. And we invade their body by placing plastic tubes and catheters into them. We disturb the microbiome by giving them lots of antibiotics. And we change the acid content in the gut by perhaps giving H2 blockers or PPIs. We aim to give them early breast milk, perhaps as buccal colostrum, but this has been collected in plastic syringes, which may also be used to deliver breast milk. The breast milk we give them has been expressed and stored in plastic bottles, or perhaps we use cup feeding. And even when we give donor milk, it's been frozen and pasteurized. So it's not surprising that the microbiome of a preterm infant is completely different to a term infant. So if we ask the question, what is the ideal microbiome of a preterm infant? We really don't know. We cannot assume that what is healthy in a term infant is necessarily helpful in a preterm infant. So we can study this, we can document the different patterns, but we really do not know what is the optimal community structure of these bacteria. And you can think of human evolution and human existence as a tug of war between, on the one hand, managing the beneficial microbes, and on the other hand, excluding harmful microbes. We need microbes in our gut for normal human existence. But equally, we don't want to become colonized with pathogens such as Shigella or E. coli. So this is the major challenge for human existence. And then consider the challenge for the newborn baby. On the one hand, they must establish tolerance for healthy bacteria, such as bifidobacteria, but on the other, activate immunity, produce IgA to exclude pathogens such as E. coli. And you might ask yourself the question, how does a 24-week gestation infant know which are the good guys and which are the bad guys? Now, many years ago, when I started my research, I assumed that milk was simply for feeding, it was simply there for nutrients and fluid, that you tip milk into this long tube called the gastrointestinal tract. You suck out the fluid and nutrients. So I thought of milk as being a food. And then at the end of the tube, you produce the waste product. So this is what I used to think milk feeding was all about. But what we've come to learn over the last 20 years is that breast milk is part of a developmentally regulated maternal-infant biochemical signaling pathway, producing lots of metabolites that control multiple processes in the body. And our research team particularly is focused on the role of the metabolites, the immune interactions, and the microbes in premature infants. So again, I wish to emphasize that nutrition is more than nutrients, and breast milk is more than food. Within the gut, interactions continually take place between the microbes, the nutrients, and the immune system. And this is true for you and me today, as well as it's true for the babies on your unit. And this interaction changes over the life course, so that the healthy pattern, the healthy interaction in an old person is quite different to that in a baby. And we can conceptualize NEC as being a breakdown in the interactions between these three elements. But what we know is that NEC is not a single disease, that it's probably the end result of lots of different pathologies. But fundamentally, these pathologies include epithelial health, levels of inflammation in the gut, potentially ischemia from compromised blood supply. And when we take a deeper look, we can think of the microbes in the intestinal lumen separated from the gut immune system by a single layer of enterocytes. And through this layer of enterocytes, dendritic cells will reach out and sample the bacteria, drag them into the lamina propria where they interact with T cells. And then there is an interaction between the gut immune system and the systemic immune system. And this relationship is very, very complex. And here is a simplified version from one of our recent papers, explaining the critical role of cell surface receptors like TLR4, whole-light receptors, and the interaction that takes place in the lamina propria with your T regulatory cells. So how do gut bacteria work? What are the mechanisms of what we call crosstalk? And how might probiotics act and reduce the risk of NEC? Well, first of all, we know that bacteria in the gut, such as probiotics, release bioactive compounds. These might be pro or anti-inflammatory. We know that bacteria may reinforce the gut barrier, but equally pathogens might damage the gut barrier, and they're likely to modulate epithelial cell function. Some of the bacteria translocate into the lamina propria where they can interact with immune elements such as T regulatory cells, but pathogens can translocate and cause disease. But there's also evidence that probiotics may, for example, stimulate IgA production. Necrotizing enterocolitis, as you know, is a terrible disease. Unfortunately, this is a pathological term that is applied to a clinical syndrome. Depending on what unit you work in, NEC probably affects up to 10% of infants born less than 28 weeks gestation. It has a multifactorial etiology, and there's no single diagnostic test. Importantly, it strikes without warning, and 50% of those with confirmed NEC develop really serious illness requiring surgery. And 50% of those who need surgery will die, and many of the survivors may have lifelong short bowel and cognitive impairment. And the fear of NEC dominates feeding practices in preterm infants. So we can think of gut health as a spectrum. And this is depicted by the iceberg here, where the tip floating above the surface is NEC, But what we don't see is everything that's happening below the surface. The differences in the microbiota between a very dysbiotic pattern compared to a rich and stable pattern. Some babies with high levels of gut inflammation, some babies with very poor metabolic or epithelial health. This is a very complex process, and much of this takes place without us being able to see it. We have no window into the gastrointestinal tract. We can do X-rays, but they really don't tell us very much about what is happening at a tissue level. So NEC is the most visible example of dysbiosis, but we should consider the continuum of gut health. What is floating below the water surface in terms of dysbiosis, inflammation, feed tolerance, growth? And here we were only really concentrating on the gut. But remember that the respiratory system and the skin have their own patterns of microbiota. So we have a challenge here to see into the dark. So all preterm infants are dysbiotic. They have a lack of diversity. They don't have very many bifidobacteria. They have a much higher presence of pathogens like Enterobacter. And the pattern that we see in babies before they develop NEC is a bit like a more extreme version of this dysbiosis. It's important to remember that microbes have functional effects, that the problems associated with dysbiosis are due to changes in the metabolites produced by the bacteria, not simply the bacteria themselves. So here's a quick cartoon showing the various factors that might be important in the development of NEC. So there may be genetic factors that make you less likely to develop NEC. There may be enteral feed components, HMOs or other components of breast milk that make your gut healthy. And you have a healthy mucus layer here depicted in yellow. You have an active, healthy epithelium, and you have good blood flow. So you have diversity, lots of healthy microbes, and few unhealthy microbes. But then as you get more dysbiosis, this may be associated with worse oxygen supply, or it may be associated with certain enteral feed components, perhaps cow milk protein antigens. And what we see is a loss of microbial diversity and an increasing presence of pathogens. And eventually this leads to inflammation in the epithelium, a breakdown of the mucus layer. And as well as the microbes, we need to remember that there are all these other metabolites depicted here by blue, red, and green dots that are impacting on gut health. So we see a change in the metabolites, the function of these bacteria. We see a thinning, disruption of the mucus layer, damage to the epithelial. And then this is what leads to necrotizing enterocolitis. And as I mentioned before, this involves activation of toll-like receptors on the cell surface, activation of cytokines, particularly IL-6 and IL-8, and a loss of the Paneth cells in the gut. So how do we make the microbiome better? Well, we can avoid doing things that make it worse, avoid the use of H2 blockers and PPIs, limit the amount of antibiotics. We can promote early colostrum, kangaroo care, and mother's fresh breast milk. But we can also consider the role of probiotics. So what are probiotics? So probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. So they're live. It's possible that component parts may have an effect. Most of these are bacteria, but they may be fungal. We have to give them in a sufficient dose. They have to be able to resist digestion by gastric acid, and they need to be able to engraft and become a permanent member of the gut bacterial community. And usually this means they require regular administration. So probiotics are very different to prebiotics. A prebiotic is a food for a probiotic. So HMOs are food for bifidobacteria. It's important to be aware of the bacterial nomenclature. At a species level, we talk about E. coli, but there may be a specific strain of E. coli that's important, for example, E. coli 0157. Staph aureus is the species, but there are certain strains of, for example, MRSA. And when we think about probiotic species, quite often we're talking about Bifidobacteria infantis, Bifidobacteria breve, Lactobacillus acidophilus, or Streptococcus thermophilus. But there are many other bacterial species. And then we need to be aware of the strains of these species. So within Bifidobacteria infantis, there's a strain EVC 001, which is available. Bifidobacteria breve BBG 001 is a specific strain. And it's really at strain level that we need to understand the mechanisms. So there are lots of potential mechanisms here. These may be at strain level. So within the same species, some strains may not work. But it's also tricks into which the probiotic is delivered. The environment for that probiotic also matters. Halved versus an infant on human milk versus an infant on formula milk, the effect might be different. So there are certain mechanisms that are common for many probiotics. These include colonization resistance, short-chain fatty acid production, regulation of intestinal transit, exclusion of pathogens, and enterocyte turnover. There are other effects that are quite common at the species level, for example, vitamin synthesis or bile salt metabolism. But then there are some rare effects that only exist at a strain level. These might be neurological, immunological, or even potentially endocrinological. So what is the mechanistic evidence in preterm infants? So here's a paper from a few years ago looking at B. infantis EVC 001 and looking at levels of enteric inflammation in healthy breastfed infants. So you see here the electron microscope of bifidobacteria. You see they have this Y shape, this bifido shape at the end of them. And what you can see here is that where there is a very high concentration of bifidobacteria, you have a lower level of calprotectin than where there is a very low concentration of bifidobacteria. And when they look at the cytokines, fecal cytokines, in grey here you see the control levels, and in green the levels where the babies have the supplemental bifido infantis. And you can see lower levels of certain cytokines in the infants who receive bifido infantis. There's also been a little bit of work in preterm babies, but not very much at all. So this study looked at babies with a mean birth weight of 1.4 kilos who received Lactobacillus reuteri compared to placebo. And the infants receiving the Lactobacillus reuteri got to full feeds fast with better weight gains. But you can see here in the graph that they had lower levels of inflammation. So babies on L. reuteri had lower levels of TNF-alpha, IL-17, IL-8. So this is some preliminary evidence the probiotics might work by modulating enteric inflammation. So there are likely to be multiple mechanisms in preterm babies. There might be metabolic. Maybe they competitively exclude other pathogens. They may reinforce the epithelium. They may have effects on gut transit or motility. So there's limited evidence from term infants and preterm infants showing that this is associated with changes in the composition of the microbiome and decreases in fetal cytokines. So do these work in the neonatal unit? Well, there have been a lot of RCTs, as you probably are aware. At least 25 to 30 different probiotic species have been studied. This leads to potentially 100 different combinations of probiotics. So how do we decide which strain is most effective? So if you considered 3 probiotic species, Bifido infantis, Bifido bifidum, and Lactoacidophilus, then the possible trials could be B. infantis versus placebo, B. bifidum versus placebo, Lactoacidophilus versus placebo, B. infantis and B. bifidum versus placebo. You get the idea. There are lots of different studies. So as part of the Espergan group, Chris van den Akker conducted a network meta-analysis. Here you see the individual effect on the risk of NEC. But this includes studies that just have 1 species or potentially 3 or 4 species. You see also here the effect on late onset sepsis. And overall, you can see that for NEC and for sepsis, most studies lie to the left of 1, i.e. suggesting that there is a beneficial effect, but very few of these studies reach significance on their own. Perhaps there's 1 study here that might be relevant. So a network meta-analysis allows you to combine this. In a standard meta-analysis, you compare treatment A versus treatment B, and you might compare treatment B against treatment C. But there's no direct trial of treatment A versus C. And a network meta-analysis allows you to compare this. So you take the direct evidence from the trials, and then you produce indirect evidence that allows you to compare A to C. So a network meta-analysis combines both of these types of evidence. And you can then expand it by including lots extra comparisons. So the statistics around this are very complicated indeed, but it allows you to produce this type of graph where you can see placebo here compared to a trial that used bifidololum and rhamnosus. And the size of the dot here tells you how many infants were in that study. And the thickness of the line tells you how many studies took place. So the thicker the line, the more comparisons you have. So that's the basis to a network meta-analysis. This is a recent Cochrane review looking at all the studies and producing a funnel plot. So what you see here is trials that reduce NEC, here trials that increase NEC. And we would expect that the same number of trials appear here, small trials appear here as here, but you can see there's a bit of an absence of studies here, which we would expect. This suggests a degree of publication bias. But look again at this dot here. This is the largest trial ever conducted. This is the UK PIPS trial that compared a bifidobacteria to placebo. And it doesn't lie at the top of the funnel, which is what you would expect. It lies directly on 1. There was no effect in this study. And this is a concern because it suggests that the effects of probiotics might be species or strain dependent. I'm not going to go into the data in total, but there are now more than 50 RCTs, including more than 10,000 babies. These show a very strong effect on reduction of NEC in the babies who get probiotics, with a relative risk of 0.54, reduction in all-cause mortality, relative risk 0.7. But in the extremely low birth weight infants, the effect is not significant because few trials included these very small babies. What's the data from observational studies? So a couple of years ago, Sanjay Patoli and colleagues looked at a meta-analysis of non-randomized studies, i.e. observational studies, and they found 30 trials, including over 75,000 infants, with a similar reduction in NEC to the randomized controlled trials, an effect on reduced sepsis, and an effect on all-cause mortality. And all of these effects were highly significant. And even when they looked at low birth weight, extremely low birth weight, they did see reduction in NEC. There was a suggestion that multi-strain products might be superior, and 3 studies did report sepsis from the bacteria. And so this is the data from the observational studies. It looks very similar to those from the randomized controlled trials. All-cause mortality is less, sepsis is left, and only low birth weight babies are also just significant here, just touching 1 for the overall effect. So we have data from 80,000 infants in controlled trials and observational data. And the strength of data to support the use of probiotics is stronger than for donor breast milk. So what's the problem? Well, a big challenge, as I say, is that the effect may exist at a strain level or species level. It's possible that all Bifido infantis are effective, or maybe it's only certain strains. It may be that you need a combination of bacteria for it to be effective. But if that's the case, which of these individual species is having the effect, and do they work synergistically? And although we can conduct a network meta-analysis, we're still limited by the original trial design and quality. You might ask if the effect is the same in breastfed and formula-fed infants. Well, in this observational study conducted in Austria, the reduction in NEC from probiotics only appears in breastfed infants. And the same was true in another study. However, other studies have shown no effect of milk type. But it is possible that the probiotic effect might be greatest in the presence of bioactive components that are only found in breast milk, such as human milk oligosaccharides. There is a lack of long-term data. We know that the early microbiome imprints, so the probiotic may have long-term effects, but few studies have explored later allergy or neurodevelopment. And it's uncertain, therefore, if early exposure is harmful or beneficial. But we should consider these in the context of less death and less NEC. And there's no good data to show any harm at present. Invasive sepsis with the probiotic bacteria, such as bifidobacteria, have been widely reported, but all of these so far have been non-fatal. But babies infected with lactobacillus do appear to be much sicker. So we need to know how to detect any contaminants or probiotic sepsis. And there was recently a death from a baby from fungus from mucormycosis in the US from contaminated probiotic products. So it's important that we use a high quality product. There is a lack of data from randomized controlled trials in the smallest infants. But if they work in larger infants, is it logical to assume they will also work in the small babies? I don't think it is. NEC is not a single disease. Consider a case of NEC in a 24-week infant on the 7th day of life after he's had just 2 days of EBM, and compare that to a 28-week infant who develops NEC after 5 weeks. We know the microbiome will be completely different in these 2 babies, and therefore the benefit of probiotic might also be very different. This is a lengthy paper from Espican looking at the requirements we might have for probiotics. We need to make sure that we have products that still have viable bacteria at the end of shelf life, that resist acid and bile digestion, and that are contamination-free. But we can also see that major deviations from what the product label says are very common. Strains are often misidentified or misclassified, and products may be contaminated. So there is an urgent need for better quality control. And when this group looked at whether the label claim is the same, they found that only 1 in 16 commercial products actually had the strains that they claim to have. Now, this might be different now, but some of these were contaminated with potential pathogens. A third of them had too few cells to actually claim any health benefit. And so the manufacturing processes and the food matrix here may be very important. So the recommendation that products should be identified to the strain level, and that drug-like products as probiotics need rigorous clinical trials. We need good systematic quality control, and we need to use certified laboratories and standardization to conduct this work. So what are the certainties? The certainties are that probiotics really do reduce NEC. They appear to improve feed tolerance, but species and strains are not equivalent. However, the safety profile is unlikely to change with more randomized controlled trials, although we might get better at identification. However, quality assurance and quality control is inadequate for most products available currently. There are lots of other uncertainties. Which strain dose should we use and when should we give it? Do we start it on the first day of life, or do we wait for the babies to have some breast milk first? And if we are to conduct further trials, should these be placebo-controlled cluster design? We know that NICUs experience cross-contamination, so a placebo-controlled trial might be quite different. And although we want to know about safety, remember that you do not detect safety for rare events in RCTs because they're not big enough. So here are 3 statements in favor of probiotics that I think are incorrect. People say that further trials are unethical and that you must use them. I do not agree with that. Our point of equipoise differs depending on the background risk and population of our NICU. People say it's simple and safe to use a commercial product. I do not think that is true. There has been recent deaths from contaminated product, and a lack of regulation exposes us as physicians and babies. People say this product works, but we know that quality control and quality assurance can be suboptimal, and the functionality may change batch to batch depending on manufacturing, transport, and delivery. However, here are 3 incorrect statements from people who disagree with the use of probiotics. They say you can't trust the meta-analysis. It is true that all trials have flaws, but many of these trials are quite high quality, and the evidence for use is better than almost any other intervention in neonatal medicine. They tell you that further trials are needed for safety, but RCTs do not determine safety. And if you want long-term follow-up, how long should we be waiting for this in the neonatal population? 5 years, 10 years? People say we should not use until regulation exists, but many countries lack access. We could be waiting years. And on the balance of probability, it seems that they cause more benefit than harm. So my own opinion is we do not know the optimal strain and dose, but the data for use are pretty convincing. I think it might be unwise to deny parents the choice unless a safe product is unavailable or your NEC rate is very low. And I do think that a multi-strain product might give the best chance of efficacy. So what can you do to help? Well, you need to help research. We need much more data. We need to collaborate better. We need to find better ways of working together using routinely available clinical data. And we need to avoid being judgmental. We need to educate. We need to educate the regulatory authorities, funding agencies, industry, parents, and our colleagues. So in summary, probiotics are beneficial for preterm infants. There are multiple mechanisms and effects. The certainty of this evidence is moderate, and more randomized controlled trials are needed. The optimal product, species, or combination is not clear. Secondly, probiotics are associated with risks in preterm infants. Invasive sepsis is rare but does occur. Contamination can be lethal, so we must only use products with good quality control. And they may be more costly, and that might be an important issue in different healthcare systems. And we need much more data. We need to conduct audits, QI, and research. We must work with parents and practice shared decision-making, both in terms of how we interpret the data, but also our clinical management and how we plan research. And I'm going to finish there. I want to thank all of the parents involved in all of our studies and the babies. I want to acknowledge all of the funders who have helped our research, and to point out that good research is a team effort. We need lots of different professionals to make this good. I'm going to take some questions after the talk, but you're welcome to visit our website here, neonatalresearch.net, or you can send me an email. So once again, thank you very much for your time and attention, and I look forward to taking the questions. So thank you for this important question about the protective role of human milk. The question is about what factors in breast milk might generate the protective effect against NEC. I think it's important to recognize that breast milk, particularly mother's own breast milk, is a very complex biological mix with hundreds of different components, including lots of growth factors and enzymes, immune elements, such as cytokines, signaling factors, insulin-like growth factor, epidermal growth factor, thyroxine, cholecystokinin. The complexity of breast milk is overwhelming, and it is impossible to know what individual factors might be most important. However, there are two components that I really would want to highlight in this regard. The first is secretory IgA. We know that this has an important role in binding pathogens within the gut and preventing them from adhering to the epithelium and translocating. We know that IgA levels in some women is much lower than in others, and that might indeed be a risk factor. Early on, IgA is produced by the infant gut, but as breast milk comes in, the secretory IgA in breast milk starts to perform this effect. But you will miss this effect in infants who are formula fed. So maybe this switch over from endogenous IgA production to secretory IgA production might occur at a time period when NEC occurs. So the first component is IgA. The second component would be human milk oligosaccharides, which I talked about. There are almost 200 different human milk oligosaccharides in human milk. These change depending on a series of factors, particularly genetic at an individual level, but we do see different patterns in different countries around the world. And in most women, around 10 or 20 human milk oligosaccharides represent the majority of all the human milk oligosaccharides. So although there are 200, in most women, 10 or 20 dominate. And we know that the incidence of NEC is related to the concentrations of one or two specific. So in our studies, the overall amount of HMO didn't matter, but the overall amount of one specific HMO called DSLNT did seem to be highly associated with the incidence of NEC. We don't really understand the mechanisms yet, but we're doing further work on it. Thank you. So this next question is about the pathophysiology of NEC, the complexity and the multitude of risk factors, including preterm birth, low birth weight, exposure to asphyxia or hypothermia, and enteral feeding. And that makes it difficult to prevent NEC with any single intervention. There is no magic bullet to the prevention of NEC. But one core component, as you highlight, is the alteration in the microbiome. So what is the concept behind the early use of probiotics? Does it make a difference when those probiotics are started? Now, this is a very important question. Again, we don't know when we should start giving the probiotics. In the large UK PIPS trial, they tried very hard to ensure that probiotics were given in the first 24 hours. Clearly, if you don't start the probiotics until day 7, there's no possibility that they could prevent NEC prior to day 7. However, I do think there's a balance to be attained here. My own preference is that babies should be given oropharyngeal colostrum, fresh colostrum, within the first few hours of birth. My clinical practice then is to continue giving only fresh mother's milk the first 24, 48 hours. I don't give donor milk in the first 1 or 2 days, and I would not give formula milk in the first 1 or 2 days. I just wait. My thinking around this is that then by day 2, day 3, there is some milk matrix or substrate in the gut with some HMOs, and that might then enable more effective use of probiotics. So typically in our unit, most babies get probiotics started around day 4. I think it's safe to do before day 4, but I wouldn't wait until week 2 because I think you miss the opportunity. And I do think that probiotic products contain very high numbers of bacteria. So I can see that if you expose the gut to a very high level of bacteria, even if it's a probiotic bacteria, on day 0, there is a possibility that that could be pro-inflammatory. So as I say, my preference is to start after we've given a bit of maternal milk, typically day 3 or day 4. But I think it would be fine to start probiotics earlier if that's what you want to do on your unit. Thank you. So this question is about dysbiosis and potentially exaggerated immune response that might be important in modulating intestinal injury, and data suggesting that prophylactic probiotics may reduce this intestinal inflammation using different types of probiotics. The question is then about thinking in inflammation, which mechanisms might be most important? So this is another really important question. It's important that we understand mechanisms in neonatal practice. And there are likely to be lots of different mechanisms here. The real challenge for us is how you can study this in preterm infants. It is possible to collect stool, feces from the infant and look at cytokines, but the techniques used for this are not widely used for preterm babies. Unfortunately, we don't really have any other way of looking at the gut. In our research group, we've looked a lot at the bacterial composition of the feces, and we can see different patterns in the babies who developed NEC. But remember that the microbiome we're studying is the feces that appear in the diaper. And really the primary site of NEC in most babies is in the small intestine. And we are not able to sample the microbiome in the small intestine. There are some inflammatory markers like calprotectin that might be helpful. However, in preterm babies, calprotectin levels are actually very high in the first few days, and after that are quite variable. So it's possible that we can look at inflammatory markers, but again, there really hasn't been very much work in these population of babies. So I agree that understanding the inflammatory mechanisms is very important here, but really we have very little data at present from preterm babies. Thank you.